we are going to start our afternoon talk. We're about to start it. And in fact, it's a real pleasure to have here Dr. Fergus Couch to talk to us today. So just to make things short, I will give you a brief introduction, and just to make sure you guys will figure out who Dr. Couch is. So Dr. Couch has contributed enormously to the characterization of genes and variants involved in the susceptibility of breast cancer using genetic epidemiology and also functional approaches. About his education, Dr. Couch received his PhD in biochemistry from University College Cork, when he did, and then he did a postdoctoral training at the Howard Hughes Medical Institute in University of Michigan, with a focus on cancer genetics. He later on moved to the University of Pennsylvania as a senior researcher in molecular genetics. Currently, Dr. Couch is a professor and chair of the Division of Experimental Pathology and Laboratory Medicine at the Mayo Clinic in the United States, with a joint appointment in the Department of Quantitative Health Science. He is a founder and leader of the Enigma Consortium that focuses on determining the clinical relevance of inherited variants in cancer predisposition genes. Dr. Couch is also co-founder of SIMBA, a consortium that has focused on the study of identifying the common genetic factors that modify risks of breast cancer among carriers of germline BRCA1 and BRCA2 mutations. In fact, Dr. Couch has published over 500 peer-reviewed articles. He has been awarded several times, and here I highlight the 2020 Outstanding Investigator Award from the National Cancer Institute for his work on breast cancer. So, Fergus, please feel free to start your talk. Thank you very much for being with us.
Thank you for the nice invitation and a really nice introduction. Okay, let me see if I can figure out how to do this. Okay. And then presentation mode. Okay, you can see that?
Yes, perfect.
Okay, good. Okay, today I will talk about predisposition genes and much of the information we've generated over the last 5 to 10 years, looking at inherited germline variants in some of the predisposition genes and their effects on cancer risk, and not just overall breast cancer, but different components, different parts of the breast cancer phenotype. So these are my disclosures. I'm required by my institution to do that. Okay, breast cancer genetics. So most people will focus on BRCA1 and 2 in the middle. These are rare in the population. Mutations are found in about 1 in 300 women with breast cancer. But the risk on the y-axis is very high, over 10-fold increased risk of breast cancer over the general population. There are other genes, such as PALB2, which is a little bit more rare, but also has high risk. And then some moderate risk genes, CHEK2, ATM, that are only 2 to 3-fold higher risk than the general population with mutations. I'll be focusing on this set of genes and a few others during this talk. Okay, so a little bit of information about breast cancer genetic testing. So around the world over the last few years, genetic testing has become very common. Generally, it's focused on women at high risk. So these are women who are diagnosed with breast cancer or ovarian cancer already, or at a young age, or have a strong family history of breast or ovarian cancer. Each country is different, but in the United States, we use a series of guidelines from the NCCN. It's a committee formed by the NCI, the National Cancer Institute, that evaluates the literature and identifies a series of guidelines for who should qualify for testing. But that leaves a lot of women in the general population. So some recent studies have shown that as many as 50% of women who carry mutations in BRCA1 and 2 do not have a family history, do not qualify as high risk for genetic testing, and so never receive testing. And so this is a real problem. It means that there are a very large number of women with mutations in these genes that are at increased risk of breast and ovarian cancer that never get tested, never get identified. So as a result of that, there's been a number of different studies that have been undertaken. And as I mentioned before, here are the guidelines focused on personal family history, multiple primaries, young age of diagnosis. These are the NCCN guidelines. Also, the American Society of Breast Surgeons have suggested testing all women with breast cancer. And one of the problems with that is that the numbers get very large, and the genetic services and the counseling services in various hospitals may not be able to handle these increased numbers. But it is a suggestion, and obviously by taking this approach, we would not miss anybody with a mutation, at least those with breast cancer. The US Preventative Services Task Force, which really focuses on women with no disease, is really, they've come up with models for testing of unaffected women, perhaps women who have a family history but don't themselves have breast or ovarian cancer. And they've come up with some risk stratifying models. So there's really many different models and guidelines that could be used to try to evaluate women for testing. But we realized back in 2017 that really there was, even though we identified the BRCA1 and BRCA2 genes back in 1994 and 1995, there still wasn't, 20 years later, there still wasn't really good information about the risks associated with each gene. We certainly understood BRCA1 and BRCA2 very well, but some of these other genes that are on this chart weren't fully understood. So we collaborated with Ambry Genetics, a genetic testing laboratory in the United States, and they provided us with results from 175,000 patients that underwent clinical genetic testing. And we did some evaluation of the risks of breast cancer based on mutations in these genes using public controls from the NOMAD group, which again is based on about 100,000 individuals. And you can see here that BRCA1 and BRCA2 are high risk. We consider high risk to be an odds ratio of greater than 4. And then there are also some moderate risk genes. Moderate risk we think of between 2 and 4. So CHEK2, ATM, maybe a few others. And then there's some other genes, BARD1 and RAD51D, which we mainly see in estrogen receptor negative breast cancer. And we'll talk about that a little bit later. But interestingly, when we did those studies, we also found a number of genes that many people thought were involved in breast cancer and were driving breast cancer. And we showed in this study that really they were not. Again, we use odds ratio of 2 as the threshold for clinical intervention. So in the United States, if you have an odds ratio of 2, you likely would qualify for MRI screening for breast cancer as well as mammography. So it has a clinical implication. So anything under 2 really does not qualify for any kind of clinical intervention. And you'll see here that we had some of the colorectal cancer, endometrial cancer, mismatch repair genes, MLH1, MSH2, PMS2, MSH6. These were all excluded as breast cancer genes. The NBN, MRE11, RAD50 complex, which is a DNA repair complex that many had thought to be involved in breast cancer, were clearly shown to not increase risks of breast cancer in a clinically relevant way. And then we had RAD51C that got fairly close to an odds ratio of 2. So based on this analysis, we couldn't really say anything about RAD51C. You can see that this and MSH6, the 95% confidence intervals overlap too. So there is the possibility in with other studies that these 2 genes might actually be moderate risk genes. And we'll talk about that a little bit later. We then took that information and looked at women that had known information about the pathology of their breast tumors. So we based this on the estrogen receptor and the HER2 markers in breast tumors. So this is essentially clinical subtypes of breast cancer, very different to molecular subtypes of breast cancer that can be defined using the PAM50 model and various other molecular approaches. So when you look at these, you find some genes that really only cause triple negative breast cancer, which is a very aggressive form of breast cancer. And some genes really mainly cause ER positive breast cancer. So some genes ER negative, some genes ER positive. We don't really understand the biology of that, but I'll show you here what I mean. If we look at ATM, ATM predominantly causes ER positive breast cancer. And you can see the three icons here with odds ratios of approximately 3 for the ER positive breast cancers, but no effect at all on triple negative breast cancer. It really does not drive ER negative or triple negative breast cancer. We know that BRCA1 causes triple negative breast cancer, a very high risk for triple negative breast cancer. And then really only, well, high risk for HER2 positive tumors, but only moderate risk for ER positive tumors. BRCA2 is a mix of all 4 subtypes, about the same, they're all clinically relevant risks for all 4 subtypes. PALB2 is the same. PALB2 influences all 4. Many people, when they read the literature, they think that PALB2 is a ER negative, triple negative gene, but in fact, it increases risk for all 4 subtypes. You can also see CHEK2. CHEK2 is mainly ER positive again. In this case, it does not cause HER2, which ATM did. It does not cause HER2 tumors or triple negative, but does cause the ER positive tumors, which many people would identify as luminal A and luminal B breast cancers. And then finally, we have RAD51C and D, that in this analysis, there were not enough numbers to study some of the subtypes, but it's fairly clear that mutations in these genes do cause triple negative breast cancer. So a lot of interesting information about the different subtypes. This information is not currently used in clinical testing or counseling of breast cancer patients, because it's very challenging to tell a patient about different subtypes. You're at risk of triple negative, but not risk of other types. That's a tough conversation with a patient. So really, this information is not currently used. We then converted that information to lifetime risks. So odds ratios are very useful, but they're mainly for statistical use and for understanding the biology. We need to convert that into the actual risk at different ages for women. So up to age 85, you can see here, if you add all the subtypes together, you can see for ATM that it's over 25% lifetime risk of breast cancer. And the same goes for these other genes. So I have the 5 most common genes shown here, and they all get over 20%. 20% is important because that in the United States, that is the number at which women would qualify for MRI screening for breast cancer as well as mammography. So again, all these patients with mutations in these genes would qualify. So having done that with the pathology-based and the overall analysis, we decided to try to look at the population-based. And again, these would be the women that don't have a family history. They develop breast cancer, but it wasn't obvious that they should get genetic testing, and many of them were not tested. So we wanted to see what were the risks, sorry for that. We wanted to see what the risks might be for women in the general population with breast cancer. So we established a carrier study. And this was a large study that was recently published in the New England Journal of Medicine. We approached a number of cohorts in the United States. So cohorts meaning that many women enroll in the study when they're unaffected at a younger age. Perhaps 100,000 women would enroll. They're followed over time. And some number of them will develop breast cancer, but they would develop many other diseases as well. So within a cohort, you can study many different diseases. In our case, we're interested in breast cancer. So we approached 10 of the largest cohort studies in the United States. And luckily, many of them had DNA samples from their patients, so we could study germline genetics. And we went ahead in my lab and screened over 70,000 women for mutations in a panel of 37 genes to try and estimate risks within the general population. So here's an outline of the study: 37 predisposition genes, over 1,300 different primer pairs, all amplified at the same time in one tube. At which point we then combine all of those primer pairs and PCR products from the one individual. We combined them from 768 different individuals and ran them down one lane of a sequencer. So high multiplexing of the samples to keep the price down, but to allow us to study all of the genes in very large numbers of individuals. This was all based on a QIAGEN amplicon panel. There's now a version 4 available. And this was custom built. We selected the genes for this panel. As you see below, we did 35,000 breast cancer patients and 39,000 matched controls. So here's what the population looks like. Again, those large numbers of cases and controls. And you see that the average age is in the 60s, right around 60 years of age for both the cases and the matched controls. That's important because if you were studying a high-risk population, these women tend to have breast cancer between 40 and 50 years of age. Those people with strong family history would often be diagnosed at that age, but we're at 60, suggesting that we are really looking at the general population. I should mention that much of our study was based on European white ancestry, so American women of European extraction, but there were large numbers of some subsets of patients, in particular the African-American population, where we had over 5,000 cases and controls, which allowed us to do a sub-study, and I'll touch on some of those results in a little while. So when we look at the frequency of mutations across the 21 main predisposition genes within the set of 37, you see here in the orange color is the frequency or the percentage of breast cancer cases that had mutations in each gene, and the black represents the unaffected women, the percentage of unaffected controls that had mutations in each gene. And you'll see that ATM, BRCA1, BRCA2, CHEK2, and PALB2, the genes I refer to as the most common genes, all had elevated frequencies of greater than 0.5%, greater than 0.5% for PALB2 and ATM, and about 1% for BRCA1, BRCA2, and CHEK2. So when we add them all together, we get approximately between 5 and 6% for all women. Again, all women with breast cancer from the general population, no selection for any family history or young age, 5 to 6% had mutations when they had breast cancer. And then when we look at controls, 1.8% of completely unaffected women matching up to those cases, 1.8% had mutations in these genes. So that tells you that 1 in 50 women, almost 2% of women walking around on the street, have mutations in these predisposition genes and are at elevated risk of breast and other cancers, which is, we did not obviously expect a number that high. It's really quite concerning, and it suggests that population testing will be needed at some point in the future. So in this population study, we then looked at these odds ratios. So how much is the risk increased? No longer looking at frequency or how common the mutation is, but now more looking at the risks. And again, we see that BRCA1 and BRCA2 are very high-risk genes. PALB2, in this case, in the general population, is only a moderate risk gene, right? Before, in the high-risk Ambry genetics data set, we had an odds ratio of greater than 4, maybe even greater than 5. In this case, it's only 3.6. So it's a little bit attenuated. But we do, and we see that ATM is lower than 2 here, which is interesting, but it does cross over 2 with its 95% confidence interval, so still clinically relevant. CHEK2 is exactly what we expected. And then we see RAD51C and D seem quite low, although there are wide confidence intervals here. Once again, the NBN gene, which, as I mentioned before, was thought to be a breast cancer gene, is completely flat, no association with increased breast cancer risk at all. So women in families that have mutations, protein truncating mutations in NBN, really are not at increased risk of breast cancer due to that mutation. Then we estimated lifetime risks. So again, age-related risks for women in the general population, again, very different to the high-risk patients. And you see here that these 3 genes all get over 20%. So people with mutations in these genes would qualify for MRI screening as well as mammography. CHEK2 the same way, but ATM here would actually fall below the 20% threshold. And so in the general population, there is some question as to whether ATM would qualify for any sort of intervention. We then looked at those 4 clinical subtypes of breast tumors, again, now in the general population rather than in the high-risk women. And again, you see the same effect that we had seen before. PALB2 causes all the different subtypes. BRCA2 causes all the different subtypes. CHEK2 causes, in this case, looks like all the different subtypes, although there's a little bit of a question about the triple negatives here that overlap with one, meaning that there's no increased risk due to the confidence interval, and ATM substantially lower for, in this case, the HER2s and the triple negatives. But generally, the results are quite consistent with what we had seen in the high-risk population, but the numbers are just a little bit lower. The risks are lower. And here's a look at directly looking at only ER-negative breast cancers, the breast cancer patients. And you can again see now with these numbers that ATM is really not doing anything for ER-negative breast cancer. BRCA1 and BRCA2, we knew. PALB2 is very high risk there as well. And interestingly, here come RAD51C and RAD51D, and they're now moderate risk genes for ER-negative breast cancer. So really they become quite important because ER-negative breast cancer is very aggressive, cannot be treated with hormonal therapy. You need chemotherapy immediately. Now it's approved for immunotherapy in breast cancer. So that's an exciting new development. But these women with mutations in these genes are at substantially increased risk of breast cancer, of that type of breast cancer. Now, moving beyond population-based versus high-risk clinic, we now decided to look at DCIS. So DCIS is ductal carcinoma in situ. Many people, it's a very early form of breast cancer. The cancer has not invaded into the surrounding tissue yet. Many people think of it as a predisposing lesion as opposed to a true tumor. Many women with DCIS will be treated by surgery and radiation and will never progress to develop invasive breast cancer, which is good news, but some of them do, quite a few of them do. So we decided to look at the high-risk population and at the population-based series for DCIS and try to figure out how common the mutations were in these different genes. And again, you see the numbers here. We have BRCA1 and BRCA2 around 1% in the high risk, along with ATM, CHEK2 and PALB2. Very similar numbers to what I had shown you before for invasive breast cancer, for overall invasive breast cancer, but now the predisposing early pre-malignant lesions that we think of as DCIS seem to have similar frequencies of mutations, which is interesting. Overall, in the high-risk population, almost 8% of women would have mutation with DCIS would have mutations in some of these genes. So the proportion of women who are at increased risk to progress to invasive cancer is quite high. And then in the population-based series, it's lower, as we've seen before for the other genes. It's lower than the high-risk people, but still about 5% have mutations in high-risk and moderate-risk predisposition genes. So when we draw that out graphically, you can see here, if you prefer graphics to numbers, you can see that, again, there's a number of important genes for DCIS. And some of them, such as CDH1, are really quite important here. We then decided to look at lobular breast cancer. So lobular breast cancer is a very different type of breast cancer. It's considered really very benign. It's in the lobules of the breast instead of the ducts of the breast where the milk is made during pregnancy and lactation. And we decided to look there. About 15% of all breast cancers are lobular in general, but they tend to be ignored for these types of studies. So we wanted to look carefully at that. And again, we were lucky enough to have both a high-risk population and a population-based series to compare the two study designs and to compare the origins of these patients. So you can see here some of the numbers. It's a very busy slide, but again, what do we find? Well, in this case, I will tell you that lobular breast cancer is mainly ER-positive breast cancer, very rarely ER-negative or triple-negative. So if you think that we're always looking at lobulars, we expect then the genes such as ATM to be important. But in fact, it wasn't, and that was a surprise to us. We thought that the lobulars would yield a high risk for ATM, but it's less than 1, which means it's not increasing risk of lobular breast cancer. And the same was true in the population-based series. However, and then we see CHEK2, similar idea, not really doing much there, and then the RAD51C and D mutations. So what gene did show up in lobular breast cancer? Were there any striking findings or important findings from the lobular breast cancer? Well, we found this CDH1 gene. Now, CDH1, for the cell biologists, CDH1 is ECadherin, and it's a very well-known marker of lobular breast cancer. In fact, it's a diagnostic test that people often will do staining of ECadherin in lobular breast cancers to show the absence of the marker. In this case, we see very high risks. You can see it's 16 to 64, very, very high risks for CDH1 in the clinical and the population-based series. So this is the highest risk driver of lobular breast cancer. But I will say that the mutations are very rare. What else did we find? Well, BRCA2, as it turns out, it looks like BRCA2 really can drive and is driving lobular breast cancer risk. It's over 4. It's considered a high-risk gene for lobular. Whereas all the other genes that we've just talked about really are not doing very much at all. They're sort of moderate risk or even no risk for lobular breast cancer. So we have to focus on BRCA2 and CDH1. And this study was recently just published about 2 weeks ago in Journal of Clinical Oncology. The next question we wanted to ask was, what about older women? Again, the genetic testing tends to be for women with early age of diagnosis of breast cancer, or those with a family history. And many of the older women over age 65 are completely ignored and never qualify for genetic testing. And in the United States, for those over 65, the only women who would ever qualify would be those with triple negative breast cancer with a family history. So a very, very small subset of those patients. So we were able to put together very large collections, 14,000 cases and 13,000 controls, and look at the mutations in these different genes in that set of individuals. And when we look at it, we find that there is still fairly substantial increased risk for breast cancer, even in women over the age of 65, when they have BRCA1 and BRCA2 mutations, CHEK2 mutations, PALB2 mutations. And interestingly, the RAD51D gene shows up. So if you have a mutation in RAD51D, which admittedly is very rare, but if you have one, you're at increased risk of breast cancer throughout your life, including when you're over age 65. And this was published in JCO earlier this year as well. We then looked at risk of ER-negative breast cancer, because again, this is a more aggressive form of breast cancer. We wanted to know how that behaved in women over age 65. So we were able to compare the ER-negatives and ER-positives and look at the risks in each set. And in the ER-positives, again, we're a little bit surprised that ATM is not doing much in those older women, whereas it was in the overall population. It was about 2.5 in the overall population, but now it's only 1.3. So not really clinically significant in ER-positive breast cancers over age 65. In contrast, CHEK2 did retain some level of clinical significance, as did PALB2 and BRCA2. And BRCA1, of course, goes away because it's predominantly causing ER-negative and triple-negative breast cancer and really is rarely seen in ER-positive. So that's why that number is a little bit lower. If we switch over to the ER-negatives, you see that the ER-negative genes jump up a lot. We have BARD1, BRCA1, BRCA2, PALB2, and again, RAD51D also showing up very, very high risks in women over age 65. So this was really a bit of a surprising finding. And then we took that information and tried to convert it into lifetime risks. Again, these are the age-related risks for the women with mutations, starting at age 65 and going up from there. So if you had a mutation in one of these genes and you survived up to age 65 without any breast cancer, what was your residual risk? What remaining risk did you have in your lifetime from 65 to death? 65 to 85 in this population is the way we modeled it. And you see that there are substantially increased risks here. They don't reach 20%, which is that number that we use for MRI screening, but both BRCA1 and BRCA2 get to 18% lifetime risk in that older population. So we argue that on the basis of these findings, that women with BRCA1 and BRCA2 mutations over age 65 should certainly be getting yearly MRIs as well as mammography. And in the United States, the rule right now is that, or the guidelines right now is that mammography can stop at age 74, I believe. But here we're saying that MRI and mammography should be used going well beyond that and up as high as age 85. ATM was a little bit lower with 10% risk, but still quite elevated in this older population, and then CHEK2 and PALB2 were about the same at about 15%. So here we have 5 genes that we think substantially increase risk of breast cancer, even in that older population. And this is really quite an important finding. And on this basis, some of the guidelines of the NCCN criteria for genetic testing have been modified to allow for more women older women with breast cancer to receive testing. Now, I mentioned earlier that we were able to do a study of African Americans. We studied 5,000 African American breast cancer cases and 5,000 matched controls. Due to some of the issues in the testing, these numbers come down to around 4,000 and 5,000 controls, 4,000 affected. And here I'm just showing you what the percentages of the mutations actually are, and then the odds ratios overall. So in the cases in the breast cancer, women with breast cancer who are African American, 7% had mutations in these predisposition genes. And that's very similar to the numbers I've shown you for the overall breast cancer high-risk women in the white, who are predominantly white, and then also in both the high risk and the population-based series. So really substantial numbers. And then in the unaffected, it's 2%, and we had 1.8% in the overall carriers population. So very similar numbers there. And what does that tell us? Let me go back to that. What does that actually tell us? Well, there's been some suggestion in the United States, people giving talks and some publications suggest that African Americans have a higher frequency of mutations in these genes, and they should be treated differently or have different testing criteria. Our finding here really shows that the frequency of mutations is very similar to those who are white. So African Americans and whites are really quite similar. And then the risks that we see associated with each of these genes are somewhat similar as well between the two populations. So the take-home message from our studies here is that, and this is the largest study ever done in the African American population for these genes, what the results suggest is that the African Americans, the current criteria can be applied to both the African American population and the white population. Risks are similar, frequency of mutations are similar. The testing panels that we use are probably quite appropriate for both populations, and we should continue the way we are. There are, of course, the possibility that other populations, Hispanic populations, Asian populations, and various other races and ethnicities from around the world there may be some populations with very different risks for mutations in each of these genes, and additional studies have to be done to evaluate those. So now I'm going to switch very quickly to variants of uncertain significance. I've been studying with Dr. Carvalho. We have some collaborations on a number of those genes at this point, and this is the work of the Enigma Consortium, and my group has done a lot of the functional studies here. So let me just take you quickly through this. A variant of uncertain significance is generally a missense variant. There are a lot of intronic variants as well, so mutations occurring out in the introns of the genes. But mainly we're focusing on missense variants that show up in some of these predisposition genes. And currently with genetic testing, still about 2 to 5% of all variants that are detected are actually variants of uncertain significance. So we know when we look at all these common predisposition genes in the ClinVar database, which is a database supported by the US government that keeps a list of all the mutations that are found by genetic testing labs, there are over 3,000 different variants of uncertain significance in each of these genes. One of the reasons for that is the genes are very large. They're huge genes, huge proteins, but still to have 3,000 different individual variants suggests that at some point in time, if we keep doing our testing the way we're doing it now, probably every nucleotide in these genes will be found to have a mutation in an individual somewhere in the world. Okay. So why are VUS problematic? Essentially, if we think about it, a patient comes in for testing, pays the money for the genetic testing, and gets a result back that says, we found a variant in your gene, but we have no idea what it means. We do not know what it means clinically. It may increase your risk, or it may do nothing at all. And so there's a lot of uncertainty with that result. The patient doesn't really know what to do. And certainly the patient does not qualify under management guidelines to receive any of these features here that I outline. The MRI screening that I've talked about before, They don't qualify for prophylactic surgery to reduce their risk of cancer because we don't even know if they have an increased risk of cancer. They just happen to have a genetic variant. And then finally, recently, PARP inhibitors have been approved as a treatment for patients with BRCA1 and BRCA2 mutations, both in the metastatic and in the adjuvant setting and neoadjuvant setting. So basically all breast cancer patients can now receive PARP inhibitors if they have a mutation in BRCA1 and 2. But of course, when you have a VUS, we don't know if that's really a risk-driving mutation, and so the women don't qualify. So it's very clear that most VUS will ultimately not be pathogenic. When we finally figure them all out, we expect that maybe 80% of them will really not be increasing risk. But that suggests that a very large number will actually increase risk. And I want to show you some of those results that we've derived from our lab. So we've been working on BRCA2. It's a very large protein of 3,400 amino acids. So almost 400 kDs in size. The main region to look at is the DNA binding domain down here. This is where all the known pathogenic, so disease-causing missense mutations are located. There really aren't others. There's one or two way down here in this PALB2 interaction domain, but nearly everything else is in this gray bar here and not anywhere else in the protein. And we have used a DRGFP reporter construct. This was a method developed by Maria Jason from Memorial Sloan Kettering in New York. She developed this a long time ago, maybe 20 years ago now. And we modified it for use in our studies in our lab. Essentially, the idea is that you have two green fluorescent protein genes in a vector. You insert this into the cell and it inserts into the genome as a stable cell line. And then you come along and you cause a cut with an enzyme. You cause a double strand break. And the cell sees this as DNA damage, as a double strand break. And it then tries to use the homologous recombination repair machinery to copy from this gene and repair the break that's been induced in this gene. If it repairs it, so if there's wild type BRCA2 activity, it will repair it, and the cell will actually then produce GFP and turn green, and we can count the number of green cells. If the mutation still disrupts BRCA2 activity, then we cannot repair the break by homologous recombination, and the cell does not turn green. So it's a very quantifiable assay. And here's an example of some of our results. There's a number of other papers that have been published, but I'm just showing this one because it has a nice graph. Across the bottom are many different variants. Vertically, on the y-axis, we're showing the number of green cells, which is the same as the amount of DNA repair activity for BRCA2. So we're starting with cells that have no BRCA2, and we're putting back in the full-length BRCA2 gene with a variant in it. And if it's wild type, you recover DNA repair activity. If it's a deleterious mutation that knocks out activity, you lose activity and you have much reduced numbers of green cells. So in the top left-hand corner, these are variants that are known by other methods to be completely neutral or benign. They do not increase risk, therefore they have wild type activity in this assay. Bottom right-hand corner, we have known pathogenic or disease-causing missense mutations in BRCA2 from family studies. So these were the standards that we used to calibrate our assay. And then we have many other variants that we've evaluated. And if you look at the black lines that run across the screen, the bottom black line means 99% probability of being deleterious. So if you're underneath the black line, there's a high probability of the variant is deleterious and perhaps pathogenic. And you can see there's quite a large number of variants there. And then above this line, it's 99% probability of being neutral or benign. So it's a highly quantitative assay. It works really well. Here's an example now from 480 different variants that we've evaluated in the DNA binding domain. We've only published about 200 of these so far. So there's some other publications in the works right now. These are two parts of the DNA binding domain. In red are the ones that knock out activity. So these are the deleterious and potentially pathogenic variants in this region. And you can see there's quite a number of them across the region. Here are the metrics or the measures for the assay. Right now, it's showing us 100% sensitivity and 100% specificity based on the known standards, the known benign and pathogenic standards that we use for this assay. So really a very, very sensitive assay, very accurate assay that can allow us to classify variants. We've also tried to move into high throughput CRISPR-Cas-based methods. So there have been some publications, in particular the Findlay et al. Paper from Leah Sterida's lab about 2 years ago in Nature, showed a very interesting CRISPR-based approach to inserting thousands of different mutations in the BRCA1 gene into the endogenous BRCA1 locus, into the endogenous BRCA1 gene in hap1 cells, haploid cells. They can then select those cells, and then they simply just measure cell survival. So what we know is that when you knock out BRCA1, cells will die. They will not proliferate. They will have cell cycle effects. They will accumulate DNA damage, and they will die over time. So essentially, when they put the library of thousands of different variants into the cells, they look for the cells that were there at the beginning and died away. And they look for the cells that were there at the beginning and are still there at high levels. If they're still there at high levels, that means that the BRCA1 mutation that was inserted into that cell was probably a wild type or a benign variant. And that's why the cells survive equivalent to the wild type. If they die away, it means that it's probably a deleterious mutation. And you can count up the number of sequencing reads from these cells and basically score each variant. So my lab is replicating a lot of that work from the BRCA1 studies in a number of other different genes. We've been studying PALB2, and there was a talk earlier in this conference about PALB2 variants and classifying PALB2 variants. So we've done about 6 different exons in PALB2. We're doing 12 different exons all across the DNA binding domain of BRCA2. We're doing large parts of RAD51C, which causes breast and ovarian cancer. And then we're doing a number of exons in ATM that cover the kinase domain and the FAT-C domain, which are the critical functional regions of ATM in the context of breast cancer. So a lot of high throughput work, a lot of high throughput sequencing studies based on CRISPR models that are ongoing at this time. One important thing here to mention is that, so here's some results. Here's an example of BRCA2 exon 18. Essentially, we use the same method of cell survival. So if the variant is deleterious, knocks out protein function, the cells will die over time. And that's why we do a day 5, a day 11, and a day 14 after introduction of the variant into the cell. The number of reads, which is what you're seeing on the vertical axis, drops down. So you can see here the stop gains. These are stop codons caused by nucleotide changes that we've inserted into the gene. Most of those variants have much lower reads than synonymous, which would be neutral variants that don't really do anything to the protein. You can see that there's a very nice separation between the neutrals and the stop codons. And when you model out this information, you find that this high throughput assay where we've studied hundreds of different variants all at the same time in one exon, it's actually giving us over 95% sensitivity and specificity for deleterious mutations. So in the orange color here, sort of pink orange color here, all of these dots are individual variants that would clearly be different from neutrals, suggesting that all of these variants here may actually be deleterious variants and may actually then be pathogenic as a result of that and disease causing. So this could really be a very useful way to study many hundreds of variants at the same time. When we've done the other BRCA2 functional studies that I showed you earlier, we've only done 480, but it's taken us about 10 years to do that work. Here, we probably have 480 in about 3 months from this experiment. So the power of this information is fantastic. But I will say that we have to validate these findings. These high throughput studies can have error in them. And every variant here really needs to be validated individually to make sure that it's a correct finding. So this is the Enigma Consortium, and we've actually formed what's called a Variant Curation Expert Panel. So for the ClinGen database that I referred to that has a list of all the mutations ever seen by clinical testing, we are now the expert panel for this, and we've got people from all over the world to join this committee and to help us make decisions and make rules about how to classify variants using the American ACMG criteria for classifying variants. So there's one panel for doing the various genes I list down below here, which is most of the predisposition genes we've been talking about today. But there's a separate panel, a BRCA1 and 2 VSEP panel, that's coming up with rules for BRCA1 and 2 classification. And I'm also a member of that. I actually chair this committee for the other genes. So the development of these rules are ongoing, and there's a number of publications in process that will come over with very clear rules using genetic pathology information, family-based information, and then functional studies where all the data can be combined together to classify variants. As an example of that, I'll show you one paper that we've recently published in the American Journal of Human Genetics. We had performed our functional BRCA2 functional study on about 154 BRCA2 VUS. And we approached Ambry Genetics, and they had observed patients with all of these variants. And they had their own ACMG classification model already developed within their company, but they had no functional studies involved. So they were using genetic data, pathology information, family data to do these classifications. And to be honest, many times they could not classify the variants. They were really only classifying about 5 or 10%. When we added in our functional data from 154 variants, you can see the result here. So before the functional data, we had 154 unclassified VUS. After adding the functional data, only 22 remained as unclassified. So we reclassified 132 variants out of 154, which is 86%. So that just shows you the power of these functional studies and how they can make a tremendous difference in this field of VUS. And here's another study. So that was with one company. We decided to then work with GeneDx, which is another testing company. We did it exactly the same way. In this case, we had studied about 100 variants. In this case, 69 variants that were VUS before any functional data. And then after we added the functional data, only 5 remained as VUS. So 5 out of 69 are unclassified, or 64 out of 69, which is over 90% of the variants ended up being classified by this method. So this shows you, at least for BRCA2, that this is a very, very powerful approach to clinical validation. And because this was done with the companies under very standardized methods, these companies can now go back to the physicians and patients who had these VUS and reclassify them and tell those patients that they're actually either benign and don't cause increased risk, or that they're pathogenic or likely pathogenic and increase their risk of breast and ovarian cancer. So it's a very powerful method. Okay, so let me summarize what we saw today. Testing of predisposition genes has become very common across the world. It's still restricted to high-risk patients, people with young age of diagnosis or with strong family histories in many countries. But that may change over time, given some of the results I showed you today about population studies and older women. It's very clear that there are large numbers of women carrying mutations of importance who are being overlooked, and hopefully changes will be made there. In the United States, many of the very large academic medical centers are already testing all breast cancer patients. So for instance, at Mayo Clinic, every breast cancer patient coming to our institution gets tested for these genes. But that's not true of all hospitals. Certainly the smaller hospitals, community practices are not doing that. And that's why the guidelines really need to be updated. I mentioned earlier that all breast cancer patients now qualify for PARP inhibitor treatment with olaparib based on the recent Olympia study. So that's really important and gives more reason why genetic testing needs to be done to identify women with these mutations, because when they develop breast cancer, they would all actually qualify for this very effective drug. We still, however, getting back to the risk, we still need population-based and family history risks for each gene. I really showed you BRCA1 and 2 and some estimates for some of the other genes, but there's some more work to be done, even in the white population. And certainly in other populations, in non-white populations, there is a huge amount of work that still needs to be done to figure out what the true risks are for women with mutations in these genes. And then finally, I showed you a lot of information about variants of uncertain significance and how can they be classified using the ACMG qualitative rules models, and then adding in the quantitative functional studies. And I gave you the example of BRCA2. And as I said, there's a lot of work now being done on all the other predisposition genes to try and use similar models of combining the functional studies with the other genetic data to resolve the meaning behind these VUS. So I'll stop there, and I will just tell you that to do this kind of work needs enormous numbers of patients. And in order to have the patients, you need collaborators from all over the world who have collected these patients or are studying various aspects of these different genes. So we have the population-based carriers project, the big group at Mayo Clinic that have helped with this sort of stuff, the variant curation expert panels, the Enigma Consortium that does the BRCA1 and 2 work, just many, many collaborators from all around the world. This is team science. This is the only way that this work can be done, and hopefully it will continue. So I will stop there and stop sharing. Turning it back to Marcelo.
Fergus, thank you very much. It's an amazing talk, and shedding light on a very sensitive problem. So congratulations, you showed us very impressive numbers. We already have questions. If you don't mind, we may take them, right?
Do you want to read them out, or should I look at them myself?
No, I can read for you.
Okay, go for it.
In fact, I have a couple questions, and I will put them in front of the other. First, in the big picture, it's right to say that we are really underestimating inheritance in cancer or in breast cancer based on your numbers. Yeah.
Certainly, we only look generally at the high-risk patients, and we're missing 50% of the mutations. There's thousands, many, many women out there in the populations that have these mutations. They are at increased risk. They may get the cancer when they're 50 or 60 or 70 years of age, but they'll never be recognized. They will develop their cancer without ever knowing that they could have had an early warning. If we tested everybody and recognized that they had a mutation before they ever got breast cancer, then you could warn them ahead of time, and there could be some intervention done to reduce their risk.
Okay, so now I'll take questions from the audience. The first one is from Dr. Andrew Sharrox.
Okay.
He says that I do not understand why some of these genes, when mutated, are more deleterious for generating particular molecular defined subtypes. Generally, the risk genes are involved in DNA repair. So are there certain genes that are more susceptible to DNA mutations than that would sit in the HER2 or ER pathways and hence affect one cell type or the other? An example.
I'm reading it here. That's okay. Fine. Yeah, that's exactly right. So one thing that people have to be clear about is that not all of the genes I'm talking about here are actually DNA repair genes. As an example, ATM and CHEK2, which cause the ER-positive breast cancer, are really cell cycle control molecules. ATM certainly is a master regulator of many different types of DNA repair, but also regulates the cell cycle, regulates P53, regulates many different pathways in the cell. CHEK2, similar, hundreds of different targets in the cell. These are master regulators of cell cycle and response to DNA damage, but not actually repair of the DNA damage. So the genes that seem to cause the ER-negative and triple-negative disease all seem to be the homologous recombination genes: BRCA1, BRCA2, PALB2, RAD51C, RAD51D, BARD1. These were the ones that had the higher risk of that type of cancer. The ones that cause the ER-positive breast cancer, CDH1, ATM, CHEK2, tend to be the more cell cycle oriented genes. That's all we know at this point. We really don't know why we get different subtypes of breast cancer as a result of that. All we know is that they are distinctly different in terms of the downstream pathways that they regulated. Beyond that, I can't give any more information. I don't know.
Okay, so I have another one from Lucas Del Monico. He says, sorry. He says that you did an excellent talk, and he was impressed about the lobular tumors have mutations in BRCA1 gene and not ATM. So the main questions, are there any genomic factors you are investigating about this? I say because clinically we know that the presence of lobular tumors increase the risk for infiltrating tumors over the years. Maybe there are lesions genetically closer to the aggressive tumors.
Yeah, I don't know. They're ER positive. They tend to be driven by loss of VCAD here, and it seems to be a very different developmental pathway for those tumors than for other tumors. They are predominantly ER positive, but yet they don't really behave like ER positive tumors. And certainly they're very nasty tumors if they become metastatic. You know, they're very, very difficult to treat at that point. They don't really respond very well to chemotherapy. So they're just a different type of tumor that we still don't understand a lot about. There have been some papers recently talking about tumor sequencing and doing a lot of tumor sequencing of lobular, very large numbers of lobular tumors. But I'm not really sure we have a clear idea of why they are different and how they are different and how they respond to therapy differently. In our case, it really seemed to be CDH1 and BRCA2 were the high-risk genes. And it was surprising that some of the other genes weren't more powerful, like ATM and CHEK2. But you know, our data is just an observation, right? We can only tell you what we saw. We don't know the underlying mechanism behind it.
So I have another question now from Vanessa Fernandes from Brazilian National Institute, Brazilian National Cancer Institute. And she's questioning, although mutations in PAL2 have a significant impact in cancer development, we have few variants already classified, especially missense variants. Do you think it's a matter of time for us to classify more variants as we have to BRCA1 and BRCA2? Still far from no at all? Or do you think we have other barriers to evaluate PAL2 variants?
Yeah, so this is a great question, and Marcelo knows all about this because he's working on this. Yeah, I mean, those of us that study PALB2, we have not found many missense variants in PALB2 that appear to be deleterious, even just knockout activity. No missense mutations in PALB2 have yet been classified as pathogenic because we don't have enough of them. We haven't seen enough families with those mutations, and we haven't been able to put enough clinical data together to use something like an ACMG rules-based model. But that work is ongoing. We are trying to collect that information to identify those. But right at this moment, I'm only aware of about 6 or 7 missense variants in PALB2 that seem to knock out activity and then would be likely pathogenic. However, with that being said, when I look at the results from some of the labs that are doing these high throughput CRISPR studies, I look at our own data, we do see some in each different exon. And I know Leah Starita showed some information at a recent meeting, so it was a public meeting, so I can disclose it. She did show small numbers of what looked like deleterious missenses in each of the different exons that she was looking at. And she was looking at the N terminus, which is a BRCA1 interacting region, and the C terminus, which is a BRCA2 interacting region. So it does seem that there are quite a few there, and there may be as many as 100 to 150 overall that will increase risk and will increase risk of breast and multiple other different kinds of cancer, like pancreatic cancer. So it's quite an important gene. It's an important exercise to actually go and find what those mutations are and which ones are the important ones. It's really work that's ongoing. I think there's a long way to go for all of us, but it's really important, yeah, because it's such a high-risk gene. Yeah.
So one last question from Gabriel Bandeira. Thank you for your outstanding presentation, Dr. Couch. Since the novel breast cancer predisposition genes are supposed to have low risk, do you think a study of breast cancer families to identify novel candidates genes is still an appropriate approach?
I think it is. We're certainly doing studies like that. We're continuing to try and find other genes that cause breast cancer. We've recently just completed a study of 10,000 high-risk patients that did not have mutations in the known genes. We did exome sequencing of them. And we have a few candidates that we're still trying to validate to see if they really are meaningful. I do know from other studies that people have done, when they find all these level RNA-based sequencing of these individuals, they're actually finding mutations that they may have overlooked. So like a large genomic rearrangement that may delete out half the gene. You won't spot that if you're just doing PCR of each exon. You'll never see that there was a huge deletion that removed half. Miss the mutations. We just missed the mutations and didn't detect them. So deeper genomic level sequencing and RNA sequencing level is important. Even with that, however, I do believe that there are a large number of families that are unexplained. And so there are ongoing studies. It may be that we won't find them by exome or whole genomes, well, at least by exomes. It could be that these are epigenetic events, so DNA-based epigenetic events, methylation events, other events like that that we have overlooked. There are some human diseases that have now been attributed to epigenetic alterations, inherited epigenetic alterations. And so whether breast cancer could be one of those phenotypes that are caused by this type of alteration remains to be seen. These are going to need to be very large studies with many thousands of patients enrolled to try and figure out what might be the drivers. So I think it is still a valuable approach. I think studying a few families, like 10 or 100 families, will really not be good enough. It needs to be thousand different families that don't have mutations in the known genes to try and explain, identify and explain some of the underlying risks that we don't really know right now. Yeah. So yeah, still a good approach, but it won't be so easy, right? I mean, the old days when we could take 100 families and find 10 BRCA1 and 2 mutations, that was easy. Now we're really looking for the really tricky stuff, the really rare stuff that's just going to be very challenging to track it down.
I'm pretty sure we are. So Fergus, thank you so much for your time, your attention, for shedding light on this specific topic for us. Hope to see you soon.
Yep. Thank you so much.
See you in Rio to visit
that would be
us.
nice. Yeah.
Maybe in a future meeting. So that's it. Thank you so much.
Thank you.